Good afternoon, everyone, and welcome to today's webinar sponsored by the NAVBO's Education Committee. I'm Enar Cuervo from the University of Illinois College of Medicine, and I will be moderating today's session. It's my pleasure to welcome our speaker, Dr. Karen Hirschi from the University of Virginia. She will present her research entitled Regulation of Endothelial Cell Specialization. We will also welcome Dr. Nicholas Chavkin, a postdoc also at the University of Virginia. He will monitor today's question. We have something new to announce, and I'm sure you've received something on your email. Our webinars and journal clubs, as well as other online activities, are now listed in the NAVBO Events app. Available exclusively to NAVBO members, be sure to download the app and keep track of NAVBO's online events. Throughout the presentation, please type your questions on the question box in the control panel. These questions will be answered at the end of the presentation. After those questions are answered, provided that there's time, attendees will be able to ask any additional questions in real time. If you have a question after the questions have been typed and read, please raise your hand by clicking on the hand icon on the left side of your control panel. You will be recognized, unmute your mic, and ask your question. This webinar is being recorded and archived at the NAVBO website for future use. So we have today Dr. Karen Hirschi. She's an adjunct professor and a member of the Cardiovascular Research Center at Yale, and also the alumni professor of cell biology at the University of Virginia School of Medicine, and a member of the Cardiovascular Research Center at UVA. The Hirschi Lab is primarily interested in vascular and hematopoietic development. They are focused on elucidating regulators of endothelial and blood cell commitment, differentiation, and specialization, as well as modulators of endothelial cell cycle state during embryonic development. Without further ado, welcome, Dr. Hirschi.
All right, thank you so much, Anar, for that introduction. And I'd like to take a minute to thank Bernadette and Sharon and all of the Education Committee at NAVBO for organizing these webinars. And also a special thank you to Bernadette for organizing so many activities that are now available to us as members of NAVBO, helping to keep us connected during these very challenging times. And also I'd like to thank ahead of time Nick for monitoring all the questions at the end of the talk. And finally, thank all of you for tuning in for this webinar. I really wish I could see all of you, and I hope that we can get together in person soon, hopefully for the annual NAVBO meeting in the fall, in October. So until then, I hope you all stay safe and well, and I hope to really see you soon. So when I get into my talk, I'm going to turn my camera off because the connection is more stable that way. So I don't think you can see me anymore, but I am here. Okay. Oops. All right. So back to me. All right. So as Anar said, today I'm going to tell you about some of the work that we've done trying to understand the regulation of endothelial cell specialization. And just as an overview of what my lab does, we've been very interested in understanding various aspects of blood vessel formation. And we've been studying this predominantly in the mouse embryo model system. And we try to understand regulators of different steps in this process and determine whether or not similar mechanistic steps occur in human development, of human endothelial cells and hematopoietic cells as well. And so we have human pluripotent stem cell models in the lab, including human ES and iPS cells, and we can generate human endothelial and hematopoietic cells that we can also then use in tissue engineering and regenerative medicine projects that we are involved in, such as this very interesting organs on a chip project that's headed up by Gordana at Columbia. And so this is just an overview of the various steps that we think are necessary for the formation of blood vessel structures. And we have been very interested in several of these aspects, including the differentiation of endothelial cells from mesodermal precursors, their specialization into subtypes that enable them to have distinct functions, and then also the acquisition of the vessel wall, which is mediated by endothelial cells that recruit mesenchymal progenitors. And when these two cell types interact, it transforms the mesenchymal progenitor into pericytes or smooth muscle cells that will make up the surrounding vessel wall. And today we're going to focus just on endothelial cell specification, and I'm specifically going to tell you about studies we've done trying to understand hemogenic endothelial cell development. And so we know in the developing mouse embryo that one of the first places where blood vessels form is in the extraembryonic yolk sac, which is shown here. And this tissue acts as a connection between the placenta and the developing embryo inside the yolk sac. And when vessels first form, they are in this capillary plexus that then has to be remodeled into a circulatory network. And this is not a weird thing that happens in the yolk sac. Vascular plexi form in as other tissues are developing in the embryo proper as well. And during this remodeling into a circulatory network is when there is specification of arterial and venous endothelial cells. And these cell types end up with different structures that enables them to have different functions. And in this yolk sac tissue, at the time when arterial and venous endothelial cell specification is happening, there is also specification of hemogenic or blood-forming endothelial cells. And so this happens first in the yolk sac, and then slightly later in development in the placenta, and then within the embryo itself in a region referred to as the aorta gonad mesonephros region, or the AGM. And we're going to talk more about that. And so this generation of blood from the endothelium, we think requires two different steps. The first is the hemogenic specification of endothelial cells that didn't previously have blood-forming activity, and then the formation of multilineage hematopoietic stem and progenitor cells from the hemogenic endothelial cells. And my lab has been most interested in this initial process, and we're trying to understand what are the unique cellular and molecular characteristics of hemogenic endothelial cells, how do they differ from endothelial cells that don't have blood-forming activity, what are the mechanisms that enable them to acquire this special characteristic and phenotype, and are the mechanisms then applicable to the specification of other endothelial cell types. And so I'll talk about some of the work that we've done, but I first want to briefly review hematopoietic development in the mouse model and show you the timeline relative to vascular development. And so in all mammals, including humans, there are two waves of blood production. And the first is referred to as primitive hematopoiesis. And this is predominantly the generation of immature erythroblasts, and it occurs only within the yolk sac tissue. And then a day later in development, definitive hematopoiesis begins, and this is the production not just of red blood cells, but also myeloid and lymphoid cells as well. And this occurs initially within the yolk sac tissue also, but then in the placenta, and then in this AGM region of the embryo. And so hemogenic endothelial cells are specified in all of these tissues, and they generate hematopoietic cells. But it's the ones that are generated in the AGM that are thought to be true hematopoietic stem cells, as opposed to multilineage progenitors that are produced in the yolk sac and the placenta. And this was discovered by taking these cells and transplanting them into lethally irradiated recipients, and the cells that form in the AGM are the only ones that are able to rescue the formation of all blood lineages, and those that form in the yolk sac and the placenta don't really form lymphoid cells very well. And so all of the hematopoietic stem and progenitor cells that form in these tissues then migrate into the fetal liver as it forms later in development, and then at last into the fetal bone marrow slightly before birth, which is about 20 days for most mouse strains. And so the formation of hemogenic endothelial cells in all of these tissues and their generation of hematopoietic stem and progenitor cells is extremely important for postnatal hematopoietic development and the process of formation of all blood cell types that we need postnatally. And so I'm going to tell you about some of the mechanisms that drive hemogenic endothelial cell development, but I first just want to show you this happening in the tissues themselves. And so this is a transgenic line that we made in collaboration with Mary Dickinson at Baylor College of Medicine. And I'm going to show you two movies. This one is hemogenic endothelial cells giving rise to blood cells in the yolk sac. And on the next slide, it's the cells in the AGM region. And in both cases, in these mice, endothelial cells are labeled with M. cherry, which is driven by an endothelial cell specific region of the FLIC1 or VEGF receptor 2 promoter. And in this case, the blood cells are labeled with the promoter from LMO2 driving GFP, and LMO2 is a transcription factor that's necessary for definitive hematopoiesis. And so when I play this movie, hopefully you will see the red endothelial cells giving rise to green blood cells, as shown here, and they pop off into circulation of this remodeling yolk sac plexus. We'll see another one giving rise here. And so the blood cells are formed from the endothelium, and they immediately enter circulation in this remodeling yolk sac tissue. And there's a slightly different process that happens within the AGM. So in this case, again, the endothelial cells are labeled with M cherry, and the blood cells with GFP, and this time driven with the epsilon globin promoter. And you'll see, as I play the movie, that the blood cells are being generated from the red endothelial cells. And in this case, in the AGM, the blood cells that form do not immediately pop off into circulation. They are generated and they divide and they form what's referred to as an intra-aortic cluster. And this niche, this cluster or this niche for these cells may in fact be one of the reasons why the progeny have different potential. So in the yolk sac, the blood cells pop right into circulation, whereas here they develop a niche that's thought to be important for their maturation. And so we and others are trying to understand how the microenvironment within these different tissues dictates the hematopoietic progeny. It may also be that the hemogenic endothelial cells themselves are molecularly different. And so we're very interested in addressing that as are other labs. And so we spent a lot of time trying to understand the phenotype of the hemogenic endothelial cells in both the yolk sac and the AGM. And by doing a lot of flow cytometry and colony forming assays, we found that the endothelial cells that have these characteristics represent hemogenic endothelial cells in both of these tissues. And so these cells express VEGF receptor 2 or FLIC1, c-KIT, which is the receptor for stem cell factor, and they do not express proteins typical of hematopoietic cells like CD45. They also have an SP or side population phenotype, and this is similar to what occurs in hematopoietic stem cells in adult bone marrow. So these cells overexpress multi-drug resistant pumps that enables them to extrude small molecules, including DNA binding dyes that can be used then for their identification and isolation via flow cytometry. And so if we isolate endothelial cells that have these collective characteristics from either the yolk sac or the AGM, and we plate them onto fibronectin, we could see that they'll form a small endothelial cell colony. And these cells have endothelial cell function in that they could take up di-acetylated LDL. They also express eNOS, which is not shown here. And then if you expose the cells to cytokines and growth factors that promote blood formation, you can see this explosion of blood colony forming activity. So this is a colony of erythrocytes. This is a colony of granulocytes and macrophages, myeloid cells. And this is a multilineage colony that contains granulocytes, erythrocytes, macrophages, and monocytes. And so very importantly, a single hemogenic endothelial cell can give rise to a multilineage colony, suggesting that these cells then give rise to multilineage hematopoietic progenitors. And so we spent a lot of time trying to understand, now that we had sort of a working definition of the cells in these tissues, we could then try and understand what are the processes that regulate their development. And we found that retinoic acid is a really important initiating factor in this process of hemogenic endothelial cell specification. And I'll tell you more about these studies. And we were very interested in retinoic acid initially because a long time ago, we were doing studies of the effects of retinoic acid on arterial venous formation. And we found that in embryos that lack active retinoic acid, and so these are mutants that have deleted retinaldehyde dehydrogenase 2, which is an enzyme that converts vitamin A or retinol from maternal circulation into active retinoic acid in the embryos. So embryos that lack this enzyme are essentially retinoic acid deficient. And in these embryos, we find that vascular development is disrupted, and this is a lethal defect. And one of the problems is that there is lack of vascular remodeling. And so a plexus will form, but is never remodeled into a circulatory network. And we found that the underlying problem in these mutants was lack of endothelial cell cycle control. And so in the absence of retinoic acid, there was reduced expression of cell cycle inhibitors, including P21 and P27. So in the absence of these regulators, the endothelial cells spent more time actively cycling and less time arrested in G1 state. And so I'm going to come back to this issue in a minute, but this was the underlying problem in these mutants. And so we wanted to know if arterial and venous specification is not happening properly, what about hemogenic specification, particularly in this yolk sac tissue? And so we looked at this using the definition of the cells that I previously explained, and we found that in these embryos that are retinoic acid deficient, there is a significant impairment in the development of hemogenic endothelial cells, and then also their generation of these multilineage hematopoietic progenitors. And interestingly, in these mutants, they could be rescued by providing exogenous retinoic acid, active retinoic acid. So they have receptors, they just cannot generate active retinoic acid themselves. And so when we do this, and we could do it in two ways, taking the embryos out and putting them in embryo culture and then giving them exogenous retinoic acid, or we could feed the pregnant mom retinoic acid in her diet, which is then transmitted to the embryos. And so we can do this either way, and we can see a significant rescue of hemogenic endothelial cell development, and then also their development of multilineage hematopoietic progenitors. And so we went on to do a number of other studies to try and understand what is retinoic acid doing and how is it promoting the formation of these hemogenic endothelial cells. And so this is a model of what is happening, particularly in the yolk sac, but the same processes happen in the AGM. And so we know in this tissue, which is fairly simple, there's a visceral endoderm layer on the outside, and below is the mesoderm, where endothelial cells and blood cells will be forming. And we know that retinaldehyde dehydrogenase 2 is expressed specifically in the endodermal layer, and it generates active retinoic acid, which then signals to a subset of endothelial cells in the mesodermal layer below that highly express retinoic acid receptors. And downstream of retinoic acid signaling is the upregulation of c-KIT, the receptor for stem cell factor, and downstream of c-KIT is the activation of Notch. And this leads to the upregulation and function of P27, which is a cell cycle inhibitor. And these steps are necessary then for the upregulation of other regulators, including RUNX1, which is a transcription factor that was found by Nancy Speck and other groups to be essential for the generation of hematopoietic stem and progenitor cells from hemogenic endothelial cells. And this process of hematopoietic stem and progenitor cell production is very complicated, and it involves a number of other pathways that different investigators have identified. And we're still trying to understand how all of these pathways are coordinately regulating this generation of hematopoietic stem and progenitor cells from the hemogenic endothelium. And my lab has been particularly interested in this initial step of hemogenic specification and trying to understand the role of P27 and cell cycle control in this process. And so I'm going to come back to this in a minute, but I also just want to say quickly that even though we have been very interested in understanding what promotes hemogenic specification, there are also important signaling pathways that limit how many hemogenic endothelial cells form. And this is really important because when there are too many hemogenic endothelial cells and too many hematopoietic stem and progenitor cells, they actually become lineage skewed, and they produce more myeloid cells than other lineages, and this causes hematopoietic defects postnatally. And so this is work that was done, we did in collaboration with Diona Casper in Stefania Nicoli's lab at Yale. And so what Diona found is that microRNA, miR223, is very important for putting the brakes on this process. And it does this through the regulation of N-glycosylation of proteins that endothelial cells express that are involved in this EHT process. And so I'm not going to say anything more about this. Deonna actually did a journal club for NAVBO in January to talk about this elegant work, and it's a very complicated study, and Deonna did a great job explaining all of the intricacies of this regulation. And so just a shout out to Deonna, who's going to be starting her own lab at Dartmouth soon, and to Stefania, our awesome collaborator at Yale. And so I highly recommend that you check out Deonna's journal club to discuss this work. Okay, so getting back to this issue of P27 and its regulation of hemogenic endothelium, I just want to show you a little bit of data from those studies. First, these data come from endothelial cells that were isolated from yolk sacs of either wild type or retinaldehyde dehydrogenase knockout mice or litter mates, and we were looking at the cell cycle distribution of these yolk sac endothelial cells at E8.5 when definitive hematopoiesis is happening. And so this is the distribution of endothelial cells in different cell cycle states in wild type situation. These are the ones that are arrested, and these are the ones actively cycling. And when we look at the endothelial cell cycle distribution in the mutants, you could see that there are less that are arrested and more actively cycling, which was similar to what we found in the phosphohistone 3 staining that I showed you earlier. And so this impairment of cell cycle control was also associated with this significant suppression of the development of hemogenic endothelial cells. And when we could express back P27 in the yolk sac endothelial cells, and we could do this using embryo culture and a lentiviral vector that expressed P27 back to wild type levels, we were able to restore cell cycle control in the endothelial cells, which wasn't that surprising, but then we also were able to rescue hemogenic endothelial cell development just by controlling cell cycle with the expression of P27. And so this was extremely interesting to us, and I just want to show you this very simple diagram of cell cycle regulators just to remind you what these proteins do. And so P21 and P27 are members of this KIP-CYP family along with P57, and they function to inhibit CDK2 or 4 and 6 in this G1 phase of cell cycle. And their activity prevents the CDKs from interacting with cyclin proteins and other proteins not shown here. And the collective function is to arrest cells in this G1 state and not allow them to transition through this G1S restriction point. So when P21 and P27 are active, the cells remain in G1. They don't enter S phase where they would undergo DNA synthesis and eventually get into M phase where they would undergo mitosis. So they stay in this G1 state. And so we wanted to know, well, if P27 in cell cycle control is so important for hemogenic specification, is the cell cycle different for these cells compared to endothelial cells that don't have any blood forming activity? And so in order to get at this question, we adopted a different mouse model, and these are the Fuchi mice. And they express two different fluorescent fusion proteins. Part of the geminin protein, which is fused to M venus, and the CDT protein, which is fused to M cherry. And so you can see in this time-lapse image taken from the previous paper that when cells first divide, neither of the reporters is expressed. But then as cells remain in G1 phase, there's an accumulation of the CDT protein, and the cell nuclei are red. And then if the cells transition into S phase, the CDT protein is quickly degraded, although they are transiently yellow, expressing both reporters. And then once the cells are in S phase and all the way until they divide, they are then expressing geminin driving M venus. And then the cells divide and then neither reporter is expressed again. And so we isolated the AGM from these Fuchi mice, and we isolated the endothelial cells in this tissue. And we used flow cytometry to figure out what is the distribution of these different cell cycle states in endothelial cells that are hemogenic versus those that are not. And so this is flow cytometry based on the red fluorescence versus green fluorescence. And when we do these studies, we found indeed that the hemogenic endothelial cells, there was a significantly higher proportion of these cells in G1 compared to non-blood forming hemogenic endothelial cells. And this was really interesting to us because stem cell biologists have found that this phase of cell cycle, G1, is really important for the commitment of embryonic stem cells into different lineages. So during this phase, there are a lot of differences in the expression of transcription factors, their accumulation, changes in chromatin structure, and other changes that are not seen in the other cell cycle states. And they also found that it's really important where in G1 phase the cells are. If they're early in G1, they're more responsive to signals that would induce mesoderm or endoderm differentiation. And if they're later in this phase, they're more responsive to ectodermal determinants. And so this G1 phase is thought to be a commitment window enabling phenotypic changes. And so what we found in our studies is G1 arrest is necessary for hemogenic specification, but it's not sufficient. So there are other factors, extrinsic factors, that are regulating the endothelial cell phenotype, just specifically within this commitment window. And so we're trying to figure out what are the factors that promote hemogenic specification when the endothelial cells are arrested in this G1 state. So we're also trying to understand the role of G1 arrest in the specialization of other endothelial cell types. And I can tell you that there is a significant role in arterial venous specification. And this work was presented by Nick, our question moderator, and he did a short talk for NAVBO just last week about this topic. And so I'm not going to present his data again, but I'm sure he'd be happy to take questions about his work. And so for the rest of the talk, I'm going to address some other questions that we had. So we had learned a lot about the regulation of hemogenic specification in the mouse model system. So we wanted to know if we could apply these insights to the generation of human hemogenic endothelial cells from either human ES or IPS cells, which would then could potentially serve as a source of human hematopoietic stem and progenitor cells for clinical therapies if this process could be properly optimized. And so we were able to generate a while ago endothelial cells from both human ES and IPS cells, but the system that we had put together was not very efficient, and it required a murine feeder layer, and the culture conditions were fairly undefined, and that wasn't really a good way to look at molecular regulation. And so we wanted to know if we can optimize a system now where we used human stem cells in a very defined system without a feeder layer and without any serum products, so that we can understand the regulation of hemogenic endothelial cell development and human endothelial cell development in these stem cell models. So this work was taken on by Jing, my former postdoc, who actually managed to graduate in May during lockdown, but she finished these studies before leaving, which was great. And in her work, she ended up adapting a stem cell culture system that was previously published from other labs. And in this system, human ES cells or IPS cells are plated in a monolayer on matrigel, and so with very defined factors and no murine feeder layer, and there's no requirement for the formation of embryoid bodies as well. And so in this culture system, the ES cells are first treated with GSK3 inhibitor, which promotes the formation of primitive streak cells, which you can see in this culture system. There is a significant decline in pluripotency markers like SOX2 very early in development. And then the primitive streak development is indicated by the expression of brachyuria or T. And then to the culture system is added basic FGF or FGF2, which promotes the formation of mesodermal progenitors. And you can see that this is reflected by the expression of HAND, which significantly increases during this time. And then to promote the formation and the propagation of endothelial cells from the mesodermal progenitors, BMP4 and VEGF-A are included. And you could see that there is a significant increase in the expression of endothelial genes like CDH5, which is V-cadherin. And there are also expression of other genes that are enriched in endothelial cells, such as FNB2 and FB4. But collectively, when we look at the profile of these cells, there's no clear mature signature of endothelial cells, and they're really what we consider to be primordial or primitive endothelial cells. And we can do this very efficiently in this system and generate about 85% of endothelial cells in this protocol. And then we wanted to know if we provide retinoic acid to these primordial endothelial cells at this stage, would we promote the formation of hemogenic endothelial cells and hematopoietic cells? And so, interestingly and luckily, we did see that there was this increase in the expression of RUNX1, which I mentioned earlier is the critical regulator of hematopoietic stem and progenitor cell development. And Jing was also noticing that there are blood cells that are being generated, and she could notice these in the supernatant of the cultures. And so she isolated the cells that were forming in this culture system, and she did cytospins and Giemsa staining to show by morphology the different types of hematopoietic cells that are formed. And then she could also gather back the adherent cells and the cells in the supernatant and stain them with antibodies that are specific for different hematopoietic lineages. And then we can quantify the proportion of the different type of hematopoietic cells that are forming in this system. And so I just want to say up front that we did not make a huge effort to try and characterize all of the hematopoietic cells that formed in this system. What we are really focused on was defining the phenotype of the cells that were generating the hematopoietic progenitors. And so in these studies, what we did was we then isolated all of the cells that formed that were CD45 negative. And so we wanted to get rid of all of the cells expressing CD45 that were already committed to a hematopoietic lineage. And when we did this and we looked at the hematopoietic potential of cells that were CD45 negative in these methylcellulose-based assays that I mentioned earlier, we could see the generation of different types of hematopoietic colonies, erythrocytes, myeloid cells, and also these multi-lineage hematopoietic colonies, including granulocytes, erythrocytes, monocytes, and macrophages. And so we wanted to further fractionate the CD45 negative cells and see if we could further define and enrich for blood-forming activity. And so from these CD45 negative cells, we then further fractionated them into cells expressing a variety of cell surface proteins that seem to be enriched in either endothelial cells or hematopoietic cells. And what we found is that those that are co-expressing CD31 and that are negative for CD45, that's where there was a high enrichment in this blood-forming activity, which was very encouraging to us because CD31 positive, CD45 negative is a flow cytometry definition of endothelial cells. And so we wanted to further fractionate this population and see if we could then further enrich for blood-forming activity. And so ultimately, I'm not going to go through all the data, but ultimately we found that we could achieve an 82-fold enrichment by adding additional markers. So VEGF receptor 2 or FLIC1 KDR in the human system, c-KIT, CD34, and the absence of D-cadherin and CD45. And so this is our working definition of human hemogenic endothelial cells in this system, and we can isolate these cells and plate them in on a single cell level into these colony forming assays and show that they can give rise to all of these different kinds of hematopoietic progenitors. And so with this definition in hand, then we wanted to know if retinoic acid is really promoting their development in our in vitro system. And so we did a similar culture assay, as I showed you earlier, where the cells at day 5 were either kept in control conditions or were treated with retinoic acid. And you can see this significant enrichment in the formation of these hemogenic endothelial cells. And then this is suppressed when we add an inhibitor of retinoic acid signaling. And so retinoic acid is promoting hemogenic endothelial cell development, but is it doing it in a manner that is dependent on cell cycle control, like we found was necessary in the mouse? And so to get at this question, we had to adopt a new embryonic stem cell line, and this is the H9 line expressing this Fuji reporter. So it's the same reporter that I showed you that is expressed in the mice, where we could determine by flow cytometry the cells in these distinct cell cycle states. And so Jing used these ES cells and subjected them to the same culture conditions that she previously defined, and then we could identify which cells are in what cell cycle state. And when we compare the hemogenic endothelial cells to endothelial cells that are forming but do not have blood forming potential, we could see that there's a significant enrichment of hemogenic endothelial cells that are in early and late G1 compared to non-blood forming endothelial cells, and then a significant decrease in those that are transitioning into S phase and undergoing active cycling. And so the next question was, well, if these cells really are in a different cell cycle state, if they're enriched in G1 like they are in vivo in the mouse, is retinoic acid functioning to promote G1 arrest of these ES-derived, human ES-derived endothelial cells, and is this necessary for their hemogenic specification? And so we did these studies where we treated, again, the cells either in control conditions with retinoic acid or with the retinoic acid inhibitor, and we find that indeed retinoic acid treatment is causing an arrest in early G1 state of the endothelial cells. And interestingly, this is not the case for this late G1 state, and there's a significant decrease in these cells that are transitioning from G1 into S phase. And then we saw this interesting increase in cells in SG2M. We're still trying to figure it out. But we were very interested in this role of retinoic acid in promoting early G1 state in the endothelial cells. So we wanted to know if this is necessary and or sufficient for this retinoic acid induction of hematopoietic specification. And so we did these studies where we again cultured the cells in the same culture system, but this time at day 5, they were treated not just with retinoic acid, but also with a CDK inhibitor. So this is a CDK4/6 inhibitor that causes cells to accumulate in late G1 state. And so in this case, we are sort of forcing the cells to not be in early G1, but to accumulate in late G1 state. And when we do this, and then we treat these cells that are now in late G1 more so than early G1, we lose this effect of retinoic acid. And no longer is retinoic acid allowed to or enabled to promote this hemogenic endothelial cell development. So we concluded from these studies that this early G1 state is necessary for this hemogenic endothelial cell formation. So then the next question is G1 state actually sufficient for this retinoic acid-induced hemogenic specification? So in these studies, we again used the CDK4/6 inhibitor to cause cells to accumulate in this late G1 state. And then we also used a CDK2 inhibitor, which causes cells to accumulate in early G1 relative to late G1, although not to the same extent as retinoic acid treatment. And so when we do this and we look at hematopoietic hemogenic endothelial cell development in the absence of retinoic acid, we see that neither early G1 or late G1 state is sufficient for the promotion of hemogenic endothelial cell formation. So we know from these studies that this early G1 state is necessary, but it's not sufficient for hemogenic specification, which is similar to what we were finding in our mouse studies. And so we're still trying to understand what are the factors downstream of retinoic acid that are causing transcriptional activity to change the endothelial cell fate toward a hematopoietic transition. And so this is our working model where we think retinoic acid is doing two things. First, it's controlling cell cycle and is promoting an early G1 state, which we think is a permissive signal, setting up a unique window of opportunity for cell signaling. But we also think that retinoic acid is changing the transcriptional activity of genes that are necessary for this transition from endothelial to hematopoietic cells. And so this is where we are now. We're still trying to pick apart the molecular mechanisms, and we've done a bunch of omic analysis where we have some nice leads. So that is it for my seminar. I just want to take a moment to thank everybody in my lab, and in particular Nick, who is helping out today, and my lab at Yale. This is Jing, who managed to graduate during a pandemic, and Yin Yu, who was helping out with the studies that were done in collaboration with Diona in Stefania's lab. And then, of course, thank you for funding to be able to do this. And hopefully we'll be able to get together again soon, post-COVID. Okay.
Thank you so much, Karen. At this point, I'll ask a question while we gather more questions. So I was wondering, how much evidence do you have of the retinoic acid effect being endothelial cell autonomous? Is it possible that is also acting on other cell types that are contributing to the endothelial cell fate specification?
Oh, let me put my camera back on. Okay.
There you are. It's
yes.
good to see you.
I'm not really technically proficient, but so yeah, that's a good question. And we showed in the mouse that it really is a cell autonomous effect, and we could do that using retinoic acid signaling mouse lines, so that we could see exactly where the signaling is happening. So we know in the murine system it is cell autonomous, and we are trying to figure that out in the stem cell model. We think it is because if we isolate endothelial cells and treat them with retinoic acid, we see similar effects. But it doesn't rule out the possibility that retinoic acid might be changing the microenvironment to some extent in the culture system.
Thank you so much. So at this point, I'm gonna let Nick handle the questions. Thank you.
Yeah, great. So we have a few questions already. So the first question comes from Kishore Wary. He has it kind of early, so I think it might be based on kind of the earlier slides. But he asked, the reduced expression of CDK inhibitors is interesting and makes sense why endothelial cells are hyperproliferative. Is this phenomena independent of VEGF and VEGFR2 signaling?
Yeah, that's a good question. So we didn't look for interactions with the VEGF signaling pathway specifically in the mouse studies. I think with, so I can't speak to that. I don't know if it changes the regulatory network in the VEGF signaling pathway. And so I guess I shouldn't really speculate about that, but thank you for the question.
Great. Okay, so then the next question comes from Jesus Gomez. And question is, great talk, impressive results. Have you tried to transplant the CD45 population? So we done any transplant experiments?
I assume he means the ones that are generated from the ES cell system.
I think so.
Yeah, so we purposely didn't go down that road. I think in order to generate hematopoietic stem cells that were transplantable, our system certainly would need to be further optimized. And other labs that have tried to do that specifically have shown that there are additional steps that are necessary once you have hemogenic endothelium and they have blood forming potential. In order to truly optimize the formation of transplantable cells, requires additional steps. And so we didn't really go down that road because we were specifically looking at the early specification events and its regulation.
Great. So another question from, and I apologize if I mispronounced this, Bronislaw Patowski. The question is, a great talk, which Notch genes and Notch ligands are first expressed in hemogenic endothelium?
So we know that, at least in the specification events, that it's Notch1 is the receptor that's important. Notch4 is also present, but it seems not to be playing a role from what we could tell. And then the ligands, we are not positively sure, but I would assume it's DLL4, which has been shown by other groups more specifically. So in our studies, we didn't dissect the specific receptor ligand interactions. We know for sure it's Notch1, and we think it's DLL4, but I can't rule out a role for JAG1 as well.
Great. Okay, next question from Leah Yin. The question is, great talk, Dr. Hirschi. How about the effect of retinoic acid on cell metabolism? Is that related to the cell cycle? Thank you.
Yes. Okay, that's a great question. So we don't know the answer, but we're extremely interested in that. And so the whole idea that the connection between metabolic state and cell cycle state is really interesting, and we're very interested in understanding how there's cooperative regulation there. We don't know the answer, but we hopefully will soon.
Yes, hopefully. So the next question is from Vicky Bouch. asks, Karen, great talk. Putting together this data with Nick's, thank you. Putting together this with my data, did the Hemi-C have arterial venous characteristics in culture? And does early G1 allow for venous, allow for venous fate mesh somehow with Hemi-C capacity?
Yeah, that's a great question. So in Nick's studies, well, Nick could probably talk more about this himself, but so we found that venous endothelial cell specification seems to be happening in early G1 also, and this is different from arterial specification, which seems to be happening in late G1. And so in the hemogenic endothelial cells that we isolated in culture, we did not see arterial venous gene expression enriched. We didn't really see those phenotypes at that time. I think it's a good question. We haven't really addressed it specifically. I mean, now that we know early G1 is important for this process for hemogenic specification and venous specification, we have to figure it out better. And so hopefully we'll be able to do that. We just don't have all the answers yet. But thank you. Vicky, hey.
All right, so next question from Patricia Demori. Yes, what is the source of the retinoic acid?
The source of the retinoic acid in the embryo?
Yeah, I think so.
Okay, so we know that the enzyme that makes retinoic acid, retinaldehyde dehydrogenase 2, is expressed in the endoderm layer of the yolk sac, also the endoderm in the embryo proper. And so that is where retinoic acid is formed. During embryonic development, the vitamin A or the retinol, the precursor to retinoic acid, is provided through maternal circulation. So I hope that, Pat's my postdoc advisor, so I hope I answer that right.
Probably. So then, oh, actually, a quick follow-up question, because that just came in. Oh, sorry, she says, thanks. Oh, is that retinoic acid acting on other cell types simultaneously?
I think they mean during development. So,
yes, this is just a follow-up question.
Okay, so we know in development, we have retinoic acid signaling mice. So the retinoic acid response element that is linked to LAC-Z. And so we could use these mice to determine exactly where retinoic acid signaling is happening in the mouse during this process. And we see that the signaling is in the endothelium, and it's not in other surrounding cell types. So in our in vitro culture system, we think also that it's the endothelial cells, that it's affecting endothelial cells in a cell autonomous way, because we can isolate endothelial cells and show similar induction of the hemogenic phenotype in the absence of other cell types. But we can't, in that system, we can't really rule out that retinoic acid has somehow influenced the other fraction of cells, which is very small, but still there, that are surrounding the endothelium. So we have to do further studies to show cell autonomous effects in vitro, but we know for sure that it's a cell autonomous effect in vivo.
Great. Okay, so moving on to the next question. This is from, again, I'm sorry, Hirschi Paletti Jandarahan. And the question is, what is the time scale in which the endothelial cell transitions from early G1 to late G1?
Yeah, that's a good question. And so, again, we don't know. You guys are having great questions, and we don't know all these answers, but we'll try to get at that using time-lapse imaging in vivo, and we hope to do that soon and be able to address that question. And so it's interesting that the G1 state is the phase of cell cycle that is the most different in different cell types. So S phase and M phase tend to be typically the same duration, but G1 and sometimes G2 are the states that are most variable. So I think what we find will be endothelial specific if we're looking specifically at endothelial cells, and it could be different for other cell types.
Yeah. Okay, so next question is from Xiaolei Liu, who asks, how do endothelial cells to hemogenic cell How EC to hemogenic stem cell transition specification events get shut down during later embryonic stages?
That's a great question too. So, so again, we don't really know, but it is very interesting that hemogenic specification occurs in different tissues during distinct windows of development. So I had mentioned that Diona's work in Stefani's lab has revealed, some factors, at least one factor that limits hemogenic specification to control the number of hemogenic endothelial cells that form. So there have to be other regulators that completely shut the program down so that this process doesn't go out of control and we end up with too many hematopoietic stem and progenitor cells, which we know if that happens during development, it causes postnatal hematopoietic defects. So I'm not sure what shuts it down, but I know that that's a really important process.
Okay, so the next question, another question from Leah Yin, who asks, how about the effect of cell cycle difference on the maturation of endothelial cells?
Yeah, okay, so that's a good question too. And so, initially we wanted to know, is cell cycle playing a role in the initial specification of event or the maintenance of the phenotype? And so Nick could probably talk to this more. We've looked at this more in the arterial venous model that Nick has been working on and found that even in adult tissues, the same distribution occurs where venous are in early G1 and arterial endothelial cells are in late G1, and that's maintained. So we think that that might be part of maintaining the phenotype of the endothelium in these distinct vessel types. But again, we're not entirely sure what mechanisms are at play.
Great. Oh, and I thought we had one more, but I think it was Xiaolei Liu clarifying that, asking about yolk sac and AGM getting shut down. But I think you answered that. Good.
Yes.
Okay. Good. Then I think that's all the questions we have from the panelists, or sorry, from the attendees.
All right, thank you so much. Thank you, Nick, for moderating the questions. Thank you, Karen, for your wonderful talk. If anyone still has a question that they didn't get to ask, you can send it to info@navbo.org, and we'll make sure that Dr. Hirschi gets it. I think I see we have a raised hand. 1 second. Mathilde Poulet has a raised hand if you want to unmute yourself and ask the question live. Mathilde, would you like to ask your question live?
Oh, I saw that she unmuted.
Maybe she clicked it by mistake. Anyone, thank you everyone for being here. I hope this was beneficial. These webinars are brought to you by the NAVBO Education Committee. Our next webinar, as you can see in Karen's screen, will be on Thursday, May 6th, entitled Regulation of Endothelial Cell Mural Cell Interaction During Development by Amber Stratman. And look for more information at the NAVBO Newsbeat. As usual, please fill the evaluation form at the end of the webinar when you receive it in a subsequent email, and let us know any topics or speakers that you may want to see in a future NAVBO webinar. Thank you so much for being with us today. Thank you, Nick, for that awesome handling of the questions. Thank you to the audience for the great questions, and thank you, Karen, for all the great science. Everyone have a wonderful day.
Thank you very much. Hope to see you all soon.